Hello, today I am going to be discussing the lion hypothesis. This hypothesis was developed in 1961 by Mary Lyon. It states that in somatic cells which contain more than one X chromosome, inactivation of all but one X chromosome occurs randomly in early development. Once inactivated, all progeny cells have the same X chromosome or chromosomes inactivated. This inactivated X chromosome is called a Barr body. This name is based upon the person who first observed Barr bodies, Murray Barr, in 1949. This does not mean that all cells within an organism containing more than one X chromosome have the same X inactivated. Since certain cells may inactivate different X chromosomes, all X chromosomes will be present in the organism, but not within the same cell. Furthermore, proteins which are encoded by both alleles will be produced, but not by the same cell. Because of this, organisms which have two X chromosomes are said to be a mosaic for the traits of the X chromosome. A very good real-life application of this is the tortoiseshell phenotype in cats. In cats, a single X-linked locus controls the presence of orange color. There are two possible alleles at this locus, X+, which produces black fur, and X0, which produces orange fur. Males may only be black or orange due to the fact that they are hemizygous. Females may be black, orange, or tortoiseshell. The tortoiseshell pattern arises from certain cells having the orange allele inactivated, leading to black fur, and certain cells having the black allele inactivated, leading to orange fur. So if we were going to clone a tortoiseshell cat, we know that the clone produced would never be the same as the original due to the random inactivation of these X chromosomes. Now we're going to consider a second trait in cats, long hair, which is dominant to short hair. We're going to be crossing a yellow short-haired female whose genotype is X0, X0, little h, and a black heterozygous long-haired male whose genotype is X plus Y, big H, little h. The two Punnett squares are shown to the right, and below is the branch diagram. And so we know that the progeny is going to be half tortoiseshell females and half yellow males, and of each of these groups, half are going to have long hair and half are going to have short hair. The overall phenotypic ratios are going to be 1/4 female tortoiseshell long hair, 1/4 female tortoiseshell short hair, 1/4 male yellow long hair, 1/4 male yellow short hair. That concludes my exam number 1 mini video. Thank you for watching.